Ah oui, ça c'est pas mal. Oui, tu peux dire ça. Voilà, et présentation de 35. On fait les questions, on essaie de faire de toute façon à midi. Et pour les questions dans la salle, on peut permettre. Par contre, comment la présenter? Ça arrive en exemple. Okay, I think it's time to start. Welcome to this new seminar in ecology of Montpellier. Just a few technical words. As usual, for those of you who are online, we are in a webinar mode, which means that your mics and your video cameras are automatically switched off. If you have any technical issue, please use the chat to report and we will try to help you. And if you have questions to our speaker at the end of the talk, please use the Q&A tab to ask your question, and then we will open your mic and your camera such that you can ask your question directly. And with this, I give you the floor, Thibault, to introduce to the speaker.
Thank you. Okay. Good morning, everyone. I'm very pleased and excited to welcome George Brown, who is a Brazilian researcher working at Embrapa, which is an equivalent to INRAE in Brazil. And he's a very good friend. And I was thinking how defining you in a few words. And I would say you are a soil ecologist, of course, but you are also a globetrotter. So, George, you started to travel the world looking at butterflies with your father when he was a kid, because your father is a very famous Brazilian entomologist. And then you did the first years of university in the US, University of Wisconsin, then Georgia. And then you started your PhD in University Pierre and Marie Curie in Paris, under the supervision of Patrick Lavelle, working on soil fauna ecology. And casually, Patrick was also my own supervisor. So we met together in Paris in the 90s. And then you did your field work in Mexico, and I did mine in Colombia. And I will not develop the parallel and quite exciting stories we both experienced in our adoptive country. This is another story. And okay, then you get back to Brazil, but you also had some other international experiences as an invited researcher in Mexico again, in Spain, in Germany, now again in France. So actually, you're working as a researcher in Embrapa Forestry in the city of Curitiba in the Paraná state in Brazil. You also teach at the Federal University of Paraná. You're teaching soil ecology, and among other responsibilities, you are also coordinator of the FAO working group on soil biodiversity. And well, you are involved in a number of international projects on soil community functional ecology. In some of them involving French institutions, such as the CESAB here in Montpellier. And you're here as a Make It fellow. You arrived in November. You will stay with us until mid-July. And that's it. I'm very excited to hear about the results you will present us on soil, fauna, and ecosystem services. And before giving you the floor, I will let Gilles present us briefly the Make It program.
So yeah, we just launched this week a call to host the next fellows. We're glad to have fellows just like George. And so in case you have any contacts you want to invite in the coming month, please let us know. I'll just present briefly in 2 minutes, so it won't be too long. Some benefits. So the main focus next year will be on governance and public policies for transition, including topics such as One Health, water reuse, and agroecology. So that could be relevant for the what used to be the LAPEX. The benefits include on a fixed space. So George has one here at CEF, also a stipend. Accommodation is provided. We will also take care directly of the travel costs and healthcare, home insurance, well, different kind of insurance that are needed, and also the visa costs in some occasions. Well, in all occasions, but yeah, it's always covered, except when they are European, because we don't need visa. Main requirements, so keep this in mind if you want to, if you have some colleagues. It could be any nationality, even French people, if they live abroad. But the tax residents must be abroad, outside France. Qualification, we will, it used to be 10 years, now it will be 5 years after PhD, and they need a contract, permanent or temporary, within the home institution. The duration will be 3 to 6 months. And of course, they have to be in Montpellier during the fellowship, because the whole idea is to create connections, to foster new or to advance already collaboration that already exists. It's built on a network of research units from the PEI, Programme d'Excellence ICIT. So all the institutions, we have Embrapa, we have CNRS, everyone. So every time the visiting fellow has a host, in this case, it's Thibault from CNRS, but it could be anyone from those institutions if they are part of Montpellier community. The next deadline is 30th of March if they want to come in September. If they want to arrive in January, we have still the end of June. And yeah, that's it. And then. So for you.
Okay, thank you very much, you. Merci à tout le monde qui est là aujourd'hui. Welcome. Thank you for being here. Those who are online, it is a pleasure to be here. Thibault was in Brazil in 2006, and sort of for a year. We met 2005, 2006. And so it's nice to be now sort of opposite end of things. Very nice to be here in France. First of all, I'd like to thank then Makit for the support and the availability that they've given us to come as a family here. And I'm here with my whole family until mid-July. And as well CNPq, which provides the support for part of my salary as well. So I'll be sharing with you today work that's involves quite a few colleagues and institutions. So I'd like to acknowledge them as well. Thibault is my main host here. Jérôme Mathieu is another important partner in this project. He's at Paris. And then there's several other institutions, local and international, involved both in Brazil and Canada. And I forgot to put the IDIF in Germany as well. So we have quite a few people involved in this project. And Just briefly what we are going to do and doing. So background, first of all, on what is soil fauna, what they do, why we should be looking at them, and why we should care about them. Some of the projects that are ongoing, finishing, and the present one that's kind of a continuation of Fauna Services 1. What are we doing? How are we doing it? What data do we have? How can we use it to actually go forward towards calculating and estimating nature's contribution to people, NCPs or ecosystem services, so via food webs. I'll be talking about that soon. Also, what I'm now calling biopedal-transfer functions and evaluation of these nature's contribution to future and opportunities to collaborate and cooperate with other colleagues here. So if any of you are interested after, please come talk to me. So recently a paper came out estimating the proportion of biodiversity that's in soils. They say it's almost 60% of life of the species in the world that live in soil. Of course, varies a lot depending on the taxa. Some taxa depend more on the soil. There's more of the species that are associated with soils like earthworms or incubates. But then there's quite a lot of diversity involved in processes and living and depending on soils for their survival, but not that many taxonomists or experts, particularly when we talk about soil animals. And so we have a long ways to go yet in terms of assessing soil biodiversity in general. Soil animals are an enormous zoo with probably over 1 million species in the world. More than 50 taxa, around 25% of all the world's known species are soil animals. And so it's impossible to be what I call in Brazil, todólogo, a generalist of everything. You can't be a taxonomist of all these taxa. So what most people do is they decide, okay, I'm going to work on ants and on earthworms, on termites, or on millipedes or whatever, and then they become specialists on that. And it also, the challenge of studying communities is that there is methods for the various fauna. So there's specialized methods developed for some of each of these. There are, of course, generalist methods, which I'll be talking about with all of their restrictions and limitations. But yes, we can study the community more as a whole, but there are quite a few challenges involved. When you start collecting these organisms and you want to classify them to species, then you run into the lack of who am I going to send it to? Who's going to identify those animals? How long is it going to take to get my results? And when I'm going to be able to use those data. So there's a huge taxonomic deficit, that's huge backlog of species to be classified. For instance, in my collection in Brazil, we have over 100 species to name still, and just starting to get to it like a little bit by little bit. So there are a few worldwide assessments for some taxa like nematodes, earthworms, springtails, but nowhere in the world has a whole community of soil animals or soil fauna actually been classified. And we can say we know that there's so many thousand species of soil animals here. So it's a big challenge, and the smaller the animal, the more we don't know about it. So for nematodes and mites, for instance, less than 20–30% of all known species have been classified, a huge number of species still to be looked at, and some of the better known species, of course, with more of the described proportions are generally those more charismatic or that people like to work with more, like earthworms, for instance. And these organisms are really hugely functionally important, and we'll go into that soon. Right now, a large number of ecosystem services or nature's contribution to people are due to the activity and presence of animals in soils. Not only is the soil a habitat for organisms, soil is also transformed by these organisms. Bioengineering or biophysical chemical engineering of these animals, particularly those that move soils, affect plant production. So plant production both positively and negatively, those that promote plant production, those that eat plants. The structure of the soil is altered and therefore affects water and air circulation through the soil, so the capacity of the soil to hold water and the amount of greenhouse gases emitted. The soil structure also and plant production will affect carbon sequestration and nutrient cycling through various food webs. And of course, Darwin's book way back in 1881 on earthworms talked about how they were affecting the preservation of historical monuments like Roman ruins or even Stonehenge, so the cultural heritage. And these are just a few of the services which are affected. And how do they do it? Well, they do it by what they eat, how much they feed, how they affect the soil. And so they affect decomposition, nutrient cycling, organic matter dynamics by what they eat, how much they digest, by the physical properties, by moving the soil. So it's aggregation, the porosity, water hydraulics. And then if they feed on plant pests or plant diseases, they can affect the plant production, plant health. And so there's various biocontrol agents, and all of these will affect ecosystem services. Now, what we know is that unfortunately, a lot of the data available in the world focuses on biodiversity or focuses on function or focuses on biomass. And when you put all of those three together, we actually see that there are very, very few studies that look at everything that you need to assess the impact of these organisms on ecosystem services. So only 0.1% of all sites in the world actually had data on biomass, on the species present or the diversity, and on functional processes at the same time for the same site. So this is sort of kind of the division of ecological research. And really, when you're starting to look at things in a more holistic view, you really need to be able to embrace all these three things at the same time to assess the impact on soil processes and soil function. So there's a huge gap here. And it's even more important in the global south or in the southern hemisphere. And you look that there's a huge lack of studies on functions and even on biomass, which shows that there tends to be a lot more done in the northern hemisphere and a lot less in the southern hemisphere. But I also call attention to an important fact for those here now that we're in the northern hemisphere. Please don't only look for papers in English. Recognize that a lot of things are published in other languages, in Portuguese and Spanish, even Chinese or whatever. So there's a lot of other stuff out there. You need to pay attention to that. It's not just Web of Science and Science Direct or whatever the other one called, Elsevier or whatever. You need to look at these other publications. And this is particularly important when you're talking about biodiversity, when you're talking about species, when you're talking about studies that are done that can contribute to global databases. And we found this, in fact, experienced it in this project, which is PI of Jean-Mathieu Nico Eisenhower, which is Fauna. And we saw that the number of transects, the number of data points went from around 3,500 to over 9,000 worldwide in just a year and a half, about a year and a half of calling for other papers, calling for other data. And the southern hemisphere here, the Neotropics, was a huge amount of data that had been published in Portuguese and Spanish, not available anywhere else, except through actually looking in these other languages. And so we managed to put these together, and we have a very rich source of data, which we now then wrote another project to look at, okay, the ecosystem services, not just the biodiversity and the function, but also the function across the different land uses in the Neotropics. So this is another project, Fauna Surfaces 1, basically, and you can see Thibault is there, Jérôme, and there's a postdoc, William, there on the right. And we've had several workshops here funded by CESAR and also FAPESP in Brazil. This project is ending now in May, and it's kind of the mother project of the one that brought me here. So we're looking at soil macrofauna. Well, what is macrofauna? It's basically the invertebrates you see with your naked eye when you're digging into the soil, when you're looking in the litter. And that involves over 50 families, over 16 orders. It's very, very diverse. And we are looking usually at their density and their biomass. And these are the main tags to go with when we're looking at our food webs. we have standardized methods for sampling these organisms. Tropical soil biology and fertility, TSBF, was a UNESCO program that started off in the 1980s. And this method's been applied all over the place now. Then it was also standardized by ISO and then by the Brazilian National Standards Organization in 2023. Basically involves taking monoliths of soil, sifting through the litter and soil layers by hand. It's quite labor intensive. Yet the data is quite useful, shows the bioindicator potential of these animals. The people who are in the field, the farmers, the land users, they recognize them, they see them, so they're valued by the local stakeholders. They do have a rapid response to changes, and furthermore, they're quite abundant. So they're easy to collect, and there are a lot of them, and so you have often very high density of biomass. You see here just from a synthesis publication of the very first data set that was available and published a few years ago, shows the difference in density from the tropics to the temperate zone. In the tropics, you have a lot of social insects, ants and termites mainly. And then if you look at biomass, the biomass is mostly of earthworms. But you see that there is quite an impact of land use on these organisms. And so we developed more recently sort of a template for this data now that's in the Soil Fauna project. And we realized that this is probably one of the most widely used methods worldwide to look at soil fauna. So macrofauna, there's manuals in several languages. It's sort of the quick and dirty method. Obviously the quick and dirty means that it's probably not perfect for the overall, for a lot of the different taxa, but it's sort of the most quick and the easiest and probably best method to look at the community, and accept the fact that it's quite labor intensive. So what we've done now is take the data for particular regions, and let's start looking at assessing then possible ecosystem service provisioning. Sort of how can these communities now be used to calculate ecosystem services that are related to sustainable development goals in this. We'll go this in just a bit now. So we focused on this project here in France. I'm focusing on Atlantic forest, a small part of the Atlantic forest in Brazil, and hopefully we'll be able to compare that with a Mediterranean hotspot here in France. And the idea is to calculate these and vast value of these ecosystem services and identify the land use management configurations that best conserve soil diversity and ecosystem services. So we're looking at these services here, basically associated with carbon and nutrients and water, and then plant production, so biological control and plant productivity. And these are the SDGs associated with that: zero hunger, good health and well-being, clean water sanitation, climate action, which has to do with the greenhouse gases, and life on land, which has to do with biodiversity. So how do we go from the data we have or the organisms to the services? So this is just a kind of a complicated, lot of things in this figure, but I'll go through it slowly here just to show how we go from what we would call moving of the soil and how it affects water infiltration, soil aeration, and aggregation. And of course, the services associated with those are water storage and soil, carbon storage, and gas exchanges, which are regulating services. And then how do we go from the feeding? And we do that through pedotransfer functions, what I'm now going to call biopedotransfer functions. And then the feeding, which has to do with what they eat and how they affect nutrient cycling and decomposition, is basically through, well, what they eat and how much they eat, which can be calculated through soil food web models. And then, of course, plant production is also affected by if they eat plants directly, or if they affect plants indirectly through either nutrient cycling or stimulating microbes that affect plant production. And of course, pollination is another great thing to look at, but we haven't been looking at it right now. So what I call the pulo do gato, which is an expression in Brazil, which would basically mean, well, how do we strategically go from one thing to another? And the pulo do gato is kind of the expression that says, oh, it's a great discovery, but it has certain risks. And in a sense, we want to avoid falling on our heads. So we have data on macrofauna and we have data on the soil, and we want to assess how do these influence functions that ultimately affect ecosystem services, and then through the human values, what we call the nature's contribution to people. Well, we can do that through various ways. We can do that from the abundance to the functions, looking at trophic networks, so food web models that tell us how much they eat, what they eat, how much they excrete, how much they die and reproduce and all that kind of stuff, and what is their turnover rates. And we can also do it 2A and B through correlations. So there are various ways you can assess soil health by using various indices, so the INVOL or the global indicator, global index of soil quality, and various other indicators that have been proposed in the literature, or through correlation tables. We can do it through exclusion and inclusion experiments, and there's quite a bit of literature out there talking about how ants, termites, earthworms, various organisms affect plant production or decomposition or nutrient mineralization, and then the pedotransfer functions, which allow us then to look at how do this activity affect processes, and then the valuation methods going from the functions to the services or nature's contribution to people through valuation methods, which I'll go into later. So here's our data set for the project Fauna Services. We have around the same very similar number of data sets from both Amazonia and Mata Atlântica, and about the same number of transects as well, a bit more in Amazon, but than the Mata Atlântica. And we have then a smaller data set from northern Paraná, which has more of the biomass values, which we'll be using to go through the food web models and some of the other calculations. And we hope to do the same thing here for France, if we can get the data for the region here in the Mediterranean hotspot. So this is just a Google Earth view of the region of northern Paraná, around 24,000 square kilometers, so about a third of the size of Occitanie. You can see very few forest fragments, just a few dark green spots, which are basically the forests left over after the 1930s, when people arrived here, and basically over the last 60, 70 years have taken out almost all the forests and left mostly crops. So the number of cropping transects reflects basically the status of this region as a very important agricultural producer. So most of our transects are from crops, a few from pastures, and a few from native vegetation. So we'll be doing those comparisons mainly between these three vegetation types, and looking at how they affect ecosystem service delivery. And of course, within the crops, we have different kinds of crops that we can also compare, and different management practices as well. So tillage, no tillage, tree crops, annual crops, etc. So our data is basically formatted like this. We have tables with transects, always at transects levels, but sometimes we have different layers, so different depths. Sometimes we have only the full monolith, so the full hole altogether. We have biomass and many times, but always tax abundance and individuals per meter squared. And we have variables related to management, so describing the sites, and around almost 20 soil variables, which allow us to then look at soil quality and the relationship between the organisms and their soil environment. So here's how we've gone through looking at the nutrient transfer, nutrient cycling, and we've taken two food web models, which were developed recently. This one in 2018 and this one a couple of years ago. And so food webs and flux web, and we've combined the two. And this way, and we've expanded the latter one, food webs to include nitrogen, phosphorus, and calcium. So here are the people involved in this. We've also had the help at the beginning with Anton Potapov and Benoit Gauzin. I don't know how to say his name, but they were involved originally, and now more recently we've been doing it through the help of Zoe and Robert at Canada. William is the postdoc of Fauna Services. Francesca is a new doctoral student involved in this effort as well from Paris. And Elisa is from Université Sorbonne, Paris. So we've used, basically what we're going to do is generate food web models, not as complex as this one, which was done by Anton Potapov for a larger community, including from microbes all the way to top predators, vertebrates. But we'll be focusing on the middle part of this food web, which is basically the insects, the arachnids, the arthropods, myriapods, and oligochaetes and mollusks. So we have 17 taxa. We'll be looking, we have data basically that we'll be using biomass to then look at assimilation, production, consumption, death, and the CNP ratios of both what they eat and of the animals themselves, and what do they proportionally feed on. So we know that not all of these animals are strictly feeding on one food source. So we have predators, we have herbivores, we have omnivores that eat whatever, the small and large decomposers, and the food sources, which are basically living plant material, the detritus and various sources, so plant litter, organic soil, organic matter, the wood, and the microbes, which are for basically the microbivores. And so we have then also the cannibals or whatever, the ones that eat themselves as well. So that's part of the model, and also in producing the nutrient fluxes. And now we've expanded these models to include nitrogen, phosphorus, and calcium as well. So here's just well, what do they eat? Well, we take this from a synthesis paper of Anton Potapov talking about the various proportions of food eaten by the various organisms from microbes to megafauna. We'll be just taking those values for the macrofauna, assuming that all the species are doing around the same thing. Of course, these are big assumptions, but we have to make them in order to get somewhere. So these are the taxa that we're looking at. And unfortunately, we haven't finished the model, and we're running through some bugs. But I think that probably by the end of this week, or at least we're already at the end of this week, by the end of next week, we'll already have some results. So I don't have anything to show you right now. But basically, it would be something like this here, this lower part. This is a Nature paper published last year by Anton Potapov and the group from data from Vietnam, looking at green and brown food webs. So the green food webs are the above ground ones. The brown food webs are the ones associated with litter and soil. And they compared rainforest and rubber plantations. And then so they have the top predators, they have vertebrates and microbes. We only have the macrofauna, so we'll only be looking at a part of this. But they found that and they were only looking at energy and carbon cycling. So they found that most of the energy was channeled in the rainforest through below ground food web, and mostly through arthropods in the soil and some arthropods in the canopy. And in the rubber plantation, it was mostly through the worms, mainly because of an invasive Brazilian worm, maybe French Guianan worm, Pontoscolex, that's off there in Indonesia, has huge biomass. And so most of the nutrient cycling carbon cycling was through worms in the rubber plantation, which highlights basically that land use is super important in regulating the food web flux, which we already knew, but in this case, quantified. So this is the kind of stuff that we hope to be able to show very soon with these food web fluxes for the different land uses, both in Brazil and France. So as we go to the second part of calculating ecosystem services, we've talked about the food webs. We go now to the part related to engineering and how the organisms affect soil properties themselves. So we have the biopedotransfer, or the biophysical engineering, as Patrick Lavelle calls it, and how is it that the organisms that are moving and affecting soil properties are affecting some of the various functions and services. And we know that by affecting aggregation and porosity, they're affecting the soil as a habitat for other organisms. They're affecting plant production. They're affecting water drainage and filtration. They're affecting water storage. They're affecting the soil erosion rates, but they're also affecting how the soil stores carbon and how much the soil emits greenhouse gases. N2O, CH2, and methane. So these are all things that need to be looked at, and we don't have data to assess all of these, unfortunately, but we do have the organisms, how much they eat, how much they assimilate, and we have that, and we can calculate that through soil food webs. We have the nutrient context of the soil. Sometimes we have the nutrient context of the structures, so we know if they have a preferential feeding, if they're cleaning on more organic matter, if they're concentrating clay in the mounds or in the casts. And we have the texture of the soil and of these structures, which allows us then to look at nutrient fluxes between soil and the biogenic structures. What is more difficult to get is the turnover rates of those structures. How long do they last? I know Thibaut did some of this in Colombia, and there's been a few other studies in the literature, which we can infer. And then what is the physical structure of those structures? So are they compact? Are they decompact? How long do they last? And that has the physical structure has a lot to do with its turnover. And we can infer that from the literature, hopefully, and hopefully we can improve some of the current carbon turnover models, century model of the Ross Sea, to include soil fauna, to then assess how is it that these organisms and their bioturbation affect carbon sequestration, carbon turnover, and greenhouse gas emissions. Finally, in terms of the plant production, we know that the organisms can either biostimulate plant production, or they can feed on the plants, either roots or shoots. So we have these that we can assess from food web models. We have the biostimulation rates from the literature, mainly for termites, ants, and earthworms. But something that's a bit more difficult to get data from is the actual interaction between these organisms when they're present. So how do they interact when they're in the food web? How do they interact with the microbes? And how they interact with soil structure? So those are challenges that we have to deal with as well, like I mentioned for the biophysical engineering. So we have these two things still to go forward. So I've now synthesized a bit how we go from the data we have, the biodiversity data, the organisms, the land use, their abundance, their biomass, their how much they feed, what they do through the fluxes, how they promote the plant growth and affect functions in terms of aggregation and structure. And now we want to go to actually assessing and valuing those services. So how do we assess what those functional rates mean in terms of services, nature's contribution to people, and how do we value those? And then how do we use those values to assess and influence land use decisions, conservation planning, and public policy? So that's number 3. That's the big challenge. I'll be talking a little bit about number 2 now. Number 2 is, okay, how do we value these services? So we made last year a major review, which is going to just be published soon, end of this month, looking at how many studies have looked at valuation. There are 238 papers in this 10-year period. Only 28 case studies actually went through valuing one or more ecosystem services. Most just talked about how they were important, blah, 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 but without actually doing the valuation properly. And most of these studies were looking at soil in a more broader sense and not so much at soil fauna, either sometimes just microbes. So we have a big gap there also in terms of addressing the value of the soil animals in terms of ecosystem service delivery. And there's 2 ways to go through the valuation process. One is sort of an easier way, and not easier, quote unquote, to quantify, which is basically the market values. So based on costs, so avoided costs, replacement costs, mitigation or restoration costs, and they're easier to relate to as a consumer. When we go to the indirect methods, which is on the right there, this is a bit more subjective. It's really variable on the audience used, so who is interviewed, where they're from, how they perceive as values. So it's a bit more subjective. and I won't be dealing with that. We'll be focusing mostly on the direct values and how that's what I call the other pulo do gato. The other challenge is going, okay, from one to the other, so from the delivery rates to the values. And we found this figure, which we modified and expanded. Doesn't look too beautiful, but off on the right, we put added climate regulation. But here it just explains to you how we can go about using the data that we have from the communities and their activity, going through, pollination, for instance, which we haven't dealt with, but it'd be great if we could. And that can be assessed through the substitution value. So if you have pollinators, how much would it cost if you lost them and you had to replace them? So if you want to plant disease suppression, we have the avoidance cost for pesticides. So how much are they affecting plant health in terms of reducing pests and diseases? So we have avoidance costs for how much it would cost to control the pest with pesticides. For the effect on nutrient cycling and on plant growth itself, either directly or indirectly through structure, fertility, etc. We have the value of nitrogen and phosphorus, for instance, that's mineralized by activity of these organisms. So we have an avoidance cost of fertilizer. So is the fertilizer value of that nitrogen and phosphorus. And finally, we go through 4 here. We have then effect on soil structure and soil aggregation, which will affect not just the water, but also the gas circulation in the soil. So as it affects carbon storage and gas exchanges, we can calculate the mitigation or the actual carbon value for sequestration. So the market value of carbon in terms of greenhouse gas emission mitigation. And we have also, finally, in 6, the replacement costs of soil. So the influence of reducing soil erosion and the influence on storage of water and infiltration. So how much would it cost to replace the water that they're making available or the soil that they're protecting? So all these sounds easy, not that easy, but these are how some of the methods that we'll be using to go from A to B. Many years ago, David Pimentel from Cornell published this paper. It's not very clear how he arrived at these numbers, but he already highlighted way back then how much proportionally the value was around 1 trillion euros per year, and what was the proportion of the value in some of these services. So he attributed most of the services due to recycling of organic matter, but then there were some others like biocontrol, pollination, and food production, actually food available, not food production, but food of invertebrates or for instance, of fungi. And then, of course, more things related to microbial activity, so biotechnology, bioremediation, nitrogen fixation, and then panogenesis, a small value, but still the value is quite significant worldwide. This is the kind of thing we would love to arrive at, to say, okay, so fauna worldwide are contributing to this much, be great if we could do that someday. Some of the values he didn't even address, which because they have sort of this subjective importance, which is like social services or more socioeconomical services related to preferences, like I like going fishing, so I use worms as a bait. I like keeping insects in my home and terraria, or I like collecting beetles like Thibault. So, you have certain values there, which you can assess by preferences or by willingness to pay. And of course, stuff related to water and sort of aggregation and soil structure he didn't address. So we have a gap there also to work forward and to work towards. We're almost done with the soil food web stuff, so we hope to be able to finish that off and send off a publication with at least the part of nitrogen, calcium, and phosphorus cycling through the soil food web. These are aspects which I still would want to work on over the next few months with colleagues here in France. So I'm looking for collaborators. If you would like to work on any of these things, please come talk to me later. So in terms of biopedotransfer functions, we'll be organizing a couple workshops to talk about this with people who are working with soil structure and soil organisms. It'd be great if we could do aspect of calculating biocontrol and pollination rates associated with soil orthopedics, the plant growth promotion, and then finally, social economists interested in nature's contribution to people. How can we value these ecosystem services, and how can we develop appropriate methods for the different land uses? So the more agricultural, the more forest, the more urban, things like that. And then finally be able to compare the results from Brazil and France. So I'm looking for collaborators. If you know of anybody or yourself are interested, it'd be great. And finally, finishing off here for the future, sort of what are some of our dreams? We know that there are global strategies, global monitoring programs that are being developed, like with the FAO, and even Soilbon is already working using soil fauna. It'd be great if we could start thinking about how can we use these data to actually facilitate the calculation and valuation of ecosystem services associated with soil and fauna. How can we think of the standardized and open repositories for these data so that they can be used and reused? And how can we use then that to bring this value back to the people who really are using the soil, so the local stakeholders, local land users, and how can we make that more easier to understand and easier to use, like cell phone apps that you can use to calculate using data that you have from the fauna and from the soil to look at ecosystem services, and how much am I getting from these organisms in my field, so that I can then go to the policymakers and policy decision makers and say, I want payments for ecosystem services. I'm protecting these organisms in my forest or in my agricultural field. I'm doing so much in terms of ecosystem service for nature's contribution to people, not just myself, but my neighbors. I'd like to get payments for this. So those will be sort of our dreams for the future. And that's it. Thank you very much. I'm open to questions.
Do we have questions in the room? Yeah.
Yeah, just one really quick question. Where is or are the Mediterranean sites that you'll be working?
Well, we have data in the global, sort of the global database, which are mainly from, I think, around Marseille and here near Montpellier. So, and they're mainly around in this region, sort of 120, 200 kilometer area around here. So it's mostly macrofauna data from colleagues that have been working on this here. So I haven't seen this data yet. I'm still working on the Brazilian data, but yeah, I think that's where it's from.
Yeah, go ahead.
Sarita, you are allowed to talk if you unmute your microphone. So maybe I can another
I
question.
can ask it for her. So we had a question from Sarita Donti. Can we use earthworms to increase fertility in the soil?
Well, yes. In fact, yes and no. We usually they are already influencing soil fertility just by being there and doing what they do, which is eating the soil and creating structures in the soil. If you want to increase earthworm contributions and their role in soil fertility, then you have the option, usually best one, is to improve the management, increase organic matter additions, which is basically food for the earthworms, and reduce soil disturbance so that you can increase your populations, and then in that way help the earthworms increase their role in the soil fertility. I usually do not propose inoculation because we don't want to be spreading common invasive species all over the world, which is already done without our help anyway. And I'm not too keen on having people, particularly in the tropics, take invasive species like the one from in Brazil that went to Indonesia, or French Guiana that went to Indonesia, and it's gone all over the world. That kind of thing we don't want to do because we usually want to avoid species invasions. So yeah, even though they're important for soil fertility, there are cases where we don't really want to be spreading and inoculating earthworms that might cause in the future, maybe not problems to your own production system, but maybe to the forest nearby or to some other natural ecosystem. So usually manage the soil in a way that you can naturally increase earthworm populations and in that way improve their role for soil fertility.
You? And do we have to
And the second one, if Rodiger
we have a
can unmute and ask a question.
Rüdiger, do you hear us?
Yes, I hear you.
Yeah.
Hear me?
Yes, that's
Yes.
good.
Okay, thank you. I just repeat my question, George. What do you think? First of all, thank you very much for this talk. What do you think about exclusion experiments to test the predictions made by the model that is in the making? Are there any already, or is it planned? Is it too costly?
Yes.
What are your ideas about it?
Thanks, Rudiger. In fact, I had a slide about that, which I removed from my presentation due to, we don't have any actual experiments planned for this particular project itself. We are mostly going to use data that's already published from these kinds of experiments, which were made in several places all over the world. And so we would like to, in a sense, have data that's more regionally relevant for depending on the place that we're using the data from. So there's data on these exclusion experiments from the United States, from Peru, from Mexico, from Martinique, from Ivory Coast, from several field experiments that were done during a big project on soil macrofauna that lasted around 12 years, led by Patrick Lavelle. And some of these results have already been published, and so they're available in the literature. And then there's, of course, field experiments and quite a few macrocosm and ecotron experiments from Europe, both here in Montpellier from the ecotron here, and also from IDIV from the ecotron in Leipzig, or not near Leipzig. So, and even in the UK, way back in Silwood Park and the Imperial College, those ecotrons as well. So there's data which is maybe not as relevant at the field level, but still relevant for some of these studies where we can then use them to infer sort of the role of these organisms and their combinations for plant growth promotion.
So the data that feed the model are actually based on measured differences that macrofauna makes compared to sites with little or no macrofauna. Is that correct?
Yeah, that's the idea. That's the idea.
Great. Yeah, good. Very good. Thank you. This was not clear to me.
Yeah. Thanks.
Thanks, George. Great seminar, learned a lot. It's not my area, but I have a question, and that is about, soil analysis, traditional soil analysis that doesn't take account of soil, fauna, and biodiversity. Is that like a real issue, or can we continue to just with conventional soil nutrient analysis? And that's
great question, George, because in fact, what people are always looking for in these, particularly policymakers and some of these big monitoring programs that are implemented all over the world is that they want to be able to take one value like carbon, organic carbon, and say, okay, I'm going to measure organic carbon, or I'm going to measure an enzyme, whatever, aerophosphatase or whatever, nitrogenase or whatever. I'm going to use that to assess everything, to tell me, about the health of the soil. And the problem is that there's a lot of factors involved in what determines, those particular attributes. And a typical soil chemical analysis, which usually gives us organic carbon, phosphorus, calcium, magnesium, available things, it gives us an idea basically of the chemical fertility. Very little tells us about the biological fertility and particularly the biodiversity aspect of that soil. It might tell us quality for crop production, but will it tell us actually about the diversity of the organisms in there? And there's some people saying that, yeah, there's some enzymes that will tell us about the microbial diversity of the soil and that have really good correlation. So, okay, great. But do they correlate with all the rest of the organisms that live in the soil, like the fauna, for instance? We don't know. So these big questions about can we use these global indicators for like carbon, organic carbon in the soil or some enzyme or microbial biomass and say, okay, we're going to use that and say it's the indicator for everything else and it's going to be like a surrogate for all the other stuff that we need to know about. Yeah, we don't have those answers yet, but everybody's looking for that, sort of like the panacea of the whole process of bioindicators is to finding that one global universal indicator, which I think is basically utopic. But that's my idea. That's my 10 cents worth.
Yeah. I have a question and thank you very much for your presentation. And I think also thank you for you and your group and your colleagues that have done all this for soil, organisms and conservation ultimately. As somebody who is working just in some of such ecosystems and actually an dynamics of soil, which is dung pads and dung beetles, we've been trying to start a conversation between us about doing these ecosystem services valuation for Mediterranean areas in France. But I haven't been so keen to go into that. And also because I don't have enough understanding on the interactions and the footwebs around and all the organisms that might be in this dung pad. So a question that I had for you is, how much complexity do you think that we must have in order to, like an idea of, do I have to have all the arthropods, all the fungi, all the plants in my ecosystem to start a conversation and start an analysis on that? And so this threshold to start doing something that could be meaningful for ecosystem services valuation. I know it's a big question.
No, it depends a lot on what your ambitions are and how and your level of if you like taking more risks or not, in a sense, because doing this for the soil food webs, just considering macrofauna is like taking a little small snapshot of a big picture, and we all would like to have the big picture, but sometimes the big picture just means that you'd need to go out and collect a whole bunch more data to be able to get that, to do that big picture. So we do it with what we have, with what we can, with what we have. And yes, we extrapolate, we infer, we do a few things, but all set up front. So these are the assumptions. This is what we are doing. And, we recognize what some of those default problems would be. And we do some sensitivity analysis or that kind of stuff where we say, okay, the range is this or this or this or this, depending on what we're looking at. And in a sense, what the main message we want to get at is that dung pads, cool, great. I mean, dung beetles, yeah, and it's just bring the message out that, okay, depending on how many dung beetles are out there, how much they represent, what is their contribution. So you need to have sort of the density, the biomass, the distribution, and you do it according to what you know about a particular ecosystem and how important they are. And then you build up, you build up from that. So I think it's a question of just go for it. You know, do it. Recognize the risk and always be honest about what your assumptions are so that people know that, you know, when you see somebody saying, oh, you know, earthworms are doing this for the world, and then you publish it in Nature, and then somebody else comes say, well, that's BS, you know, that's not true, you know, and then somebody else goes and publishes another paper saying, you know, it wasn't like that necessarily. But, you know, there's people who love to take big risks. I'm not one of those. I like to take calculated risks. But yeah, you know, it depends on what you want to do and how far you want to go with that. Go ahead, go for it. Go for it.
Thank you for your great presentation. I think you mentioned something like soil structure turnover. Could you please define that and tell me if there's a method to measure it today?
Ooh, yeah. What we do in terms of calculating the roles of soil fauna, for instance, and the soil and how they affect the soil structure is basically through thin sections. So you have impregnate the soil, and then you would assess, you know, how many of certain types of aggregates and what do they, you know, so you have images analysis. You can do that also through tomography, more expensive, but maybe easier. And then the thin sections take a lot of work as also. But you can also do more quick and dirty methods, which is doing blocks in the field that you break up, and that so that there's this sort of these methods developed for field work where you look at sort of the what is it called? Well, the FAO was promoting this, and in Brazil, there's quite a few people that do this kind of method, which basically breaking by hand the structure and then assessing the types of aggregates and the aggregate blocks and rounding them. So it's this kind of method that's based on data obtained in the field, and then of course we're talking about pictures. So in terms of turnover, it's always a difficult process because we don't know how long these structures last.
Okay. So there's no really way to.
You go to the field a whole bunch of times and do it over and over and over and over and over again. But nobody has the money for that. Experiments.
Yeah. Such a destructive method cannot let you see the temporal evolution of the process.
Yeah, what people have done is sometimes they bring the blocks to the lab, and then you scan them repeatedly, and then you have some turnover, but it's so expensive, so difficult, you know, and running those softwares and doing that. There's very few people that can actually do that. Some of them are here in Avignon. So Capoeiras, for instance, and some others, de Bayon, Claire de Bayon in Switzerland. And so there's a few groups that do that, but yeah, it's not easy. You have to have the tomographer, basically.
Other questions? No? Fine. So thank you again.